Okay, so welcome to this next video in which we are discussing activatory point mutations of KRAS. Okay, so far what we have seen is that if you stimulate a cell with growth factor, what happens is that turns the monomeric G protein called KRAS into the on state where it's bound to GTP. Downstream of that, what happens is when KRAS is in its on state bound to GTP, it causes the increased activity of MYC and C-phos-C-Jun heterodimers, which trigger a cell to move from interphase into G1 phase and begin the process of division. Right, so let's go back to our KRAS. Our KRAS is here. In its GTP, with its GTP bound to it, it's in the on state, and with GDP bound to it, it's in the off state. Now, basically, KRAS has a portion of it, which I'll denote as this portion here. There is a portion of KRAS which is the intrinsic GTPase. So KRAS has part of its structure is an intrinsic GTPase, which basically has the role of chopping this GTP down into a GDP. We're breaking it down from GTP to GDP, so chopping the terminal phosphate off of that GTP and returning it back to GDP. So, basically, when growth factor comes along, it will trigger transiently the activation of SOS, which then turns this KRAS in the off state into the KRAS in the on state. Then, after a certain amount of time, the intrinsic GTPase will return you from the on state back to the off state. So this protein has a way of turning itself off, basically. Now, if you get a point mutation in the gene for KRAS, let me show you this. So let's say we have our gene for KRAS here. Okay, so the gene for KRAS consists of loads of organic bases. So let's say this is the KRAS gene here. So this is some chromosome here, and this is the KRAS gene, and I don't actually know what chromosome KRAS is on, but it's not important for the argument. Here is the KRAS gene on some chromosome, and basically it consists of, loads and loads of organic bases, and basically what is going to happen is one of these organic bases may well change. That's what I mean by a point mutation. So let's say some mutation happens to this gene, and one of the organic bases changes. Now, the codon that organic base was in, remember when genes are transcribed into RNA, you get a complementary piece of mRNA, and then when you actually translate the mRNA, the way it works is that you don't look at every organic base on its own. Instead, you look at a triplet of organic bases known as a codon, and basically by the combination of three organic bases you have here, you will decide what amino acid to put in. So that gives you enough different combinations to have a different combination for every single amino acid, basically. Okay, so when you change one of these organic bases, so let's say this organic base here has changed in this codon, then now that codon has changed. So this codon here has changed, which means that the organic base that is going to correspond to this codon may well change. Now, it's not necessarily it's not completely necessary that it will change. The reason being that if we think about the number of codons there are, well, there are 4 options for the organic base that can be in position 1. Then for each of those 4 options, there are 4 options for what can be in position 2. And then finally, there's 4 options for what can be in position 3. So it's 64 possible codons that you can make, basically. Now, there are only around 20 amino acids. So basically, we've got far too many codons. So some codons well, some amino acids are going to have multiple codons which all code for them, basically. So it is possible that if you just change one of these organic bases, you might have changed the codon, but that both of those codons, the original codon and the later codon, actually code for the same amino acid. That's known as degeneracy of the amino acid code or of the genetic code. Degeneracy. So the amino acid genetic code is degenerate. Okay, so it's not necessarily obvious that the amino acid will change that corresponds to that codon, but often it will. Okay, so if it doesn't change, then it's known as a silent mutation because it actually produces no effect because the protein you're going to produce is exactly the same. Now, if it does change, if the organic base, the amino acid that you now put into the protein does change, then it's called a missense mutation because you have put in the wrong amino acid, so you've made a mistake, basically. So that's a missense mutation. So let's say we got a missense mutation in KRAS, so that one of the amino acids in the polypeptide is wrong. Well, basically what can happen is this one amino acid being wrong can inactivate that intrinsic GTPase. So basically, the intrinsic GTPase no longer works in the KRAS protein, and then what happens is once KRAS has got GTP bound to it, it never turns off. It just remains on forever and ever and ever. So it continues activating the BRAF kinase enzyme, which then continues to activate MEK. So MEK is always on. MEK then activates MAP kinase. So MAP kinase is always on. MAP kinase then leads to the CFOS and CJUN and MYC transcription factors always being on. And that means you're always triggering the cell to divide, so you can produce over-division of the cell. And that's how the KRAS gene can become an oncogene by a single point mutation. So you can get a gain-of-function mutation here from a single point mutation. So in the previous video, in Burkitt's lymphoma, we saw how we could have chromosomal translocations where we translocate a gene so that it's now in front of a different in front of a different promoter region, and that promoter region, if that's more active, you're going to increase the expression. We saw how that could cause a gain-of-function mutation. Now we're seeing how a single point mutation could actually produce you a gain-of-function mutation. Okay, so KRAS is a good example of how a single point mutation can lead to a gain of function of that protein. So the proto-oncogene has gone to an oncogene. The protein having too much function is then going to lead to oncogenesis.